of contraction. So what it's a rhythmic contraction. So what's the opposite of contraction? Relaxation, right? So it's contraction and relaxation. What relaxes? What relaxes is the muscles. So when food is in the mouth and mastication is completed, food must be swallowed. Then this process of peristalsis is going to allow the food bolus to move from the mouth through the pharynx. Okay, so you see this is the pharynx, it's the throat here. And by the way, I just spoke about, I was showing you exactly what I have, but we have it on the diagram class. We have got the mouth cavity. We have our mouth cavity, and then there's a mouth here. I spoke about salivary glands. What do? They secrete saliva. But saliva mechanically is just to moisten the food. There's more that we can speak about when we now go to chemical digestion. For now, we are good. We just know that there's saliva in the mouth, right? And then this young man is not showing us the teeth, but they are there, and the tongue is not showing. So when food is swallowed, the food is gonna move from here's the mouth, there's the movement through the pharynx. You see the movement through the pharynx. And what process is allowing this food to move? It's now peristalsis, that rhythm of muscles, circular and longitudinal. Circular muscles, they are in circles. Longitudinal ones are the ones that are running opposite circular muscles. So as they alternate, they are relaxation and contraction. The food bolus moves from the mouth down through the pharynx, and there goes the food to the esophagus. The esophagus, you remember, you have learned about it last time, is that long tube that is going to lead to the stomach. So all the way from the mouth, learners, to the stomach, we have got mechanical digestion. Peristalsis is allowing the food that was chewed in the masticated in the mouth. This sounding very good. Use the terminology. Masticated in the mouth, then move. The food moves through peristalsis process. It's a process, right? And now the food is going to touch base the stomach. In the stomach, we are now going to have continuous again movement, which is we call stomach churning. So the food is thrown from one end of the stomach wall to the other end of the stomach wall. Never confuse yourself, class. Yes, we do have chemical digestion in the stomach, but for now we're talking about physical digestion, which is mechanical digestion. I hope up to now you are fine. But for your information, we are talking about the entire digestive system. So from the stomach, we do have this vital organ, which is the liver. We are going to look at a big role that is played by the liver later on. And we have got the gallbladder. There's the gallbladder. Yes, it's a very close friend to the liver. We spoke about the stomach. You see the stomach, it has got different linings. We have got the pancreas, this pancreas here. You've learned about the pancreas being the endocrine and the exocrine gland. Yes, you've learned about that it is endocrine and it's also exocrine. Do you remember the definitions of the two? Exocrine is the presence of ducts, because we have what we call pancreatic ducts that are going to take the pancreatic juice. Now, this is chemical digestion. The pancreatic juice, which is secreted in the pancreas, through the ducts, the pancreatic juices are going to be now taken to the intestines, right? We have got also the endocrine part of the pancreas. You'll learn about this in your next lesson. However, just to give you a snippet of what it is all about, the endocrine part is that this pancreas secretes what we call hormones. We'll touch base later in our lesson about hormones when we speak about the functioning of the liver. So the pancreas also secretes hormones, And these hormones, now they are secreted directly into the blood. We do not have any ducts. This is just something that you must keep at the back of your mind, because this forms part of your matric syllabus. Once you have a good foundation, you will not struggle in matric, especially with the pancreas story that I was explaining to you. Very interesting. You will enjoy the lesson thereof. We also, as a revision, we have the small intestines. Small intestines, so we have got the first part, which is from the stomach, which is the duodenum. I'm not going to speak about the sphincters that we have on the entrance of the stomach and on the exit of the stomach. We have done that last time. So we've got the duodenum, which is the part of the small intestine. We have jejunum, jejunum, and we have ileum. The three, they form part of the small intestines. Later, we'll look at the significance. For now, just keep this at the back of your mind. Can you see how the small intestines are? Look at this. Look at this. There's a reason for this. We will talk about the reason later. We also have got the large intestines, which is also called the colon. So we have the ascending colon. Ascending means this is going up, right? And we have got the transverse colon. There's the transverse one that is going across, and we've got the descending one. Maybe what we need to do so that we're able to see properly, I'm going to clean up a bit. Let's clean up a bit. Finally, good, we're back. Let's clean up a bit here so that you are able to, there we go, right, there we go, right. Just for you to see the large intestines or the colon, this is the ascending colon, so it's going up, that's the ascending colon, and the transverse, that is like going across, and this continues now going down. This continues as the descending colon. Ascending is going up, transverse is going this way, and descending is going down. Very important for you to note is that once we reach the descending colon, this leads to the rectum. This leads to the rectum. Here's the rectum, and now we have got the last exit point of our undigested food material, which is the anus. If you check here by the ascending colon, you'll see this small loop here. You don't have to worry about it. This is an appendix. It doesn't play a role basically in the human's body, but it can be very painful. I had an experience with it, and it had to be removed. The pain is too much. But once they remove, you are fine. I'm up and running again. I'm a happy life science teacher. Yippee! Let us now look at, we are recapping once more, chemical digestion. So with chemical digestion, now this is where we've got enzymes. Enzymes. Do you remember enzymes? Yes. They are chemical agents. What do? They help to speed up chemical processes in the body, but they do not form part of the end product. What they do, they assist in the process. You have learned about this in grade 10. So this is definitely some kind of revision. However, I'm not very interested in the definition and more stories about what enzymes are. But we are looking at the roles played by different enzymes in our bodies, what these enzymes do. Let us check. So we've got different food substances, right? Who can mention some of them? We've got proteins, yes. What else do we have? We have carbohydrates. And what else do we have? We've got fats. These are practical things. These are things that we know about, right? And chemically, in order for them to be in simpler forms where they can be easily broken down, we need enzymes. Hence, we're talking about chemical digestion. So whether peristalsis takes place, whether stomach churning is taking place in the stomach, which is mechanical, physical, even if I stop chewing, I've got food in my stomach, but I stop chewing in the mouth, which is mechanical digestion, chemical digestion will still take place in our bodies. And this is involuntary. You like it or not, it's going to happen. You know, if we had control on this, I will decide, hey, I've got a long trip. Let me eat today. I'm going to hold to digestion. It must not happen so that I am able to sustain what is in my stomach. But guess what? You eat, you travel. After some time, the whole process in just in a second place, you ate your snacks or you ate your lunch, and now food is graciously getting digested. Later in the afternoon, if you left in the morning, you start feeling hungry because there's a need for you to refill. Amazing. Our bodies are just amazing. Trust me, they are. So now back to the business of the day. We are looking at different food types and the names or types of enzymes that help to mechanically break down these food types. So we've got proteins, right? So we've got protein molecule, and the protein molecule is made up of many amino acids, right? So we've got many amino acids. So you can safely say that the building blocks of proteins is amino acids. Again, in grade 12, you learn about my amino acids that are going to be grouped up to form required proteins in our bodies, because the proteins that are there, they are all different, right? So check this out. We have an enzyme. Be very careful on the name of enzyme responsible for breaking down specific type of molecules. We have an enzyme which is called protease. Protease. Can you really go wrong? Protein, protease. Can you see the link? You need to be smart in life sciences because you don't have to close your eyes and memorize everything. Some things they relate, they give you clues. Then you are able to say, oh, what is in that examination? What's the name of that enzyme, by the way, that breaks down protein molecules? No, it has to have okay, it's protease. Good, you got it right. That's excellent. And you can see that a protease, what it does, it breaks down the protein molecules, right? So the protein molecules are broken down. And what do we have at the end of the day? We are now going to have, now the end product that we have here is the amino acids, right? Oh, look, here we've got our amino acids. Can you see that they were a long chain, right? That was a protein, and the enzyme protease, what it did, it broke down the protein molecule, and at the end we've got amino acids. Good. However, I wanted to check something as well. So let's just quickly erase here. Very good. So let's erase here. Right. There we go. Good. I just want to direct you to something quickly. Right. So what we have here is, okay, my pen. Right, let's see if we can get it right. Right, so what we have here is the chemical digestion, and I want to, let me just write on my board. I'm just checking on my pen. Right, there we go. Right, so I was still explaining to you that amino acids and product of a protein molecule that was broken down. And what we have, just check this out. Protease as an enzyme. So we come and check. So this is the protease, and this is an enzyme. Right. Check where this is produced. So it's secreted in the stomach, right? also in the pancreatic juices, and this is also secreted in the intestinal juices, because the intestines also, they secrete their own juices. So we have got the stomach. Just know the location, the place where these different enzymes are produced. We have got the stomach, we've got pancreatic juices. I explained to you just now that when the pancreas itself secretes pancreatic juices, they are transported through the pancreatic duct to the relevant place, to the intestines, to aid, to help with the process of a protein digestion. So once we have this amino acid, this is now the simpler form of our proteins. Good. We're done. Then we move to starch molecules. When we speak about starch molecules, learners, we are talking about carbohydrates, right? So we're talking about carbohydrates here. Carbohydrates. So with carbohydrates, they are made up of many glucose molecules. We said proteins are made up of many amino acids molecules. But now carbohydrates or starch molecules are made up of many glucose molecules. And what is the name of the enzyme responsible for breaking down these starch molecules or carbohydrates? Now we have got a name of an enzyme, and this enzyme is called carbohydrates. We spoke about protease, then carbohydrates. It's the name of the enzyme. What is the function? It breaks down carbohydrate molecules. Just look at the end product. So when carbohydrates are broken down, they result now in what? In glucose. This is the simpler form of our carbohydrate digestion. Can we check as well? Where do we get these carbohydrates? Which parts of our digestive or human digestive system do we have the secretions of these enzymes? So carbohydrates, so let's move. We have got carbohydrates, and carbohydrates is secreted. It's the saliva. Definitely, you know saliva. It's secreted by the salivary glands. Where? In the mouth. So the saliva that is secreted, as I speak, I can feel that the saliva that is being produced. When you smell something nice, you salivate. So you can see that, you can basically feel, let's say you eat something that is sour, you feel the salivary glands pulling, secreting in a very obvious way saliva. In the saliva, we have carbohydrates, which is an enzyme. So going back to when we started, I said to you that in the stomach we do have mastication, which is mechanical digestion. So as a person is chewing, there is breaking down. It's a physical way of breaking down of food. However, because there's saliva that is helping to form the food bolus, the saliva contains this enzyme carbohydrates. So in the mouth we have got mechanical digestion, because as saliva contains carbohydrates enzymes will be chemically digesting or breaking down carbohydrates into simpler form, which is glucose. So I said to you, it's saliva that contains enzyme carbohydrates, but also we have got pancreatic juices and we have got intestinal juices. Look at something common between the protease and the carbohydrates. The two enzymes, the one for breaking down proteins and the other one that is for breaking down carbohydrates. I just want us to look at the similarities. So let's check quickly. If we check here, so I just want you to see the significance in terms of the parts, right? Okay, we're good. Okay, right? So I'm just going to indicate to you now what is going to happen as we have got the basic significance of friendship that we have with the parts of the bodies and the secretions that we have thereof. Never mind, my pen is, I think the ink is getting finished, but we are good. Right. You know what we can do? Let us go for a very short ad break. We'll be back in 2 minutes. Run, get some water, drink, and I'm just going to quickly go and have my new pen. All right. So, okay, never mind. I'm going to show you that we do have the common things that we have here is the pancreatic juice, and we've got carbohydrates and proteins. Same location of secretion. And we have intestinal juice and intestinal juice secreting the two enzymes. And you now look at saliva, and saliva is just a different location for the carbohydrates as an enzyme, and then protease is secreted in the stomach. Let us now look at the fat, breaking down of fats, right? So when fats must be broken down, learners, we have got a fat molecule that is made up of fatty acid and glycerol molecule. The glycerol molecule, fatty acids, right? So you see this is glycerol with fatty acids, but this must also be broken down. And what is going to help to break this down? We have an enzyme that is called lipase, which breaks down fat molecules. But look at the end product of these fat molecules. The fat molecules that we have now, the outcome is going to be glycerol and fatty acids, right? Glycerol and fatty acids, good. We have our ink. Happy. Look at the fat molecule. There's the fat molecule, right? And the fat molecule is made up of fatty acids and glycerol molecules. You see, we have got glycerol and fatty acids. They make up the fat molecule. And the enzyme called lipase, what it does, it breaks down. Lipase breaks down the fat molecule to become the products now are glycerol and fatty acids. We're speaking about fat molecule. End product is fatty acids and glycerol. It is very important for you learners to know as we do digestion that the end product of protein is amino acids, end product of starch or carbohydrates, it's glucose, and the end product of a fat molecule, when it's broken down, the end product is going to be glycerol and fatty acid. But let us look at the location where the secretion is taking place. So lipase, you see this is lipase. Lipase is secreted by the pancreatic juice and also intestinal juices. Pancreatic juices and intestinal juices. That is where we find the enzyme lipase. Look at the similarities. The only difference that we have on this table is carbohydrates, which is also secreted also, because we've got pancreatic juices and intestinal juices. The same thing applies to the protease, pancreatic juices, and intestinal juices. look at lipase, it's pancreatic juices and intestinal juices. Should you be found with a question or facing a question of where we get this name of carbohydrates or proteins or lipase, close your eyes, forget about the saliva and stomach, unless if the question is specifically asking you to address it. You know, or pancreatic juices, they have all the 3 enzymes, or they contain the 3 enzymes, and also intestinal juices, they contain the 3 enzymes. We are good. Once you know this, mechanical digestion, chemical digestion, there's no reason for you to go wrong. You are good to go. Can we look at the structural adaptation of small intestines? Structural adaptation of the small intestines. Do you remember the different sections of the small intestines? And do you remember when I showed you that look at how they are? They look like they've been packed together, right? So we are now looking at the structural adaptation. What makes the small intestines to be structurally adapted? Structural adaptation, this is not something new. You are familiar with it. If we ask, this is a general question. If we say that's what is the structural adaptation or suitability of basketball players, they must be tall. They must be tall. So the small intestines are structurally adapted. So in other words, the way they are in such a way that they are able to carry specific functions that must be performed by the small intestines. I posed a question now to say, do you still remember the 3 parts of the small intestine? Who can tell? Duodenum, yes. And what else? Jejunum and ileum. We are good. There are 3. The colon part, it has to do with the large intestines. Let us look at something interesting here. The small intestines, they have got folds, you see. It's a lot. Oh, this is overwhelming. This is a lot. You can see we have got a lot of folds, right? We've got a lot. You see, all this, it's so many, right? And what is so many? We're talking about villi, because soluble nutrients are mainly absorbed through the villi. Villi is a plural form of villus. So one villus but many villi. So what we have here, this is just a simple diagram showing us so much that is happening in our bodies. These folds, imagine your small intestines, they do have these villi. Mine too, I cannot run away from that. I'm a human being. Digestion takes place. So I also have these many folds. And each villus contains microvilli, right? And the purpose is to further increase the surface area. I want us to look at the further part. So in other words, when we've got the small intestines have got villi, and the presence of villi is to increase the surface area. To increase the surface area. But because now furthermore, from what we see here, all these millions of villi, we have got further, we've got microvilli. I'll show you, there's a beautiful diagram that is going to show you the presence of the microvilli, right? As much as we are not able to really see with our naked eyes, but we do have the folds that are found on each villus, each one of them, from the millions that we have. And they further increase the surface area. So surface area generally is increased by the presence of villi. However, further increase of the surface area And why should we have an increased surface area? There is a reason for that. That we must have an increased surface area because small intestines, they play a big, big role in the processes of digestion and the other processes that complete human nutrition. Do you remember I said we have these folds? It's a lot. The folds are many. Right, look. Look how the small intestines are folded. Look. Have a look at this. Just look. Never mind about the large intestines. We're not here. We're not worried about the ascending colon. We're not worried about the transverse colon. We're not worried about the descending colon. But we are looking at the small intestines. The small intestines, they have got a large surface area to absorb. Now, we start tapping into the significance of the large surface area. I told you that the large surface area is brought by the presence of what? Of the folds that are called the villi. So you can relate already from what we looked at previously that we do have villi, which are folds that are there to increase the surface area. So they've got large surface area. Okay, small intestine has a large surface area. The reason is to do what? To absorb. Hence, we look at what we call absorption. We'll talk a little bit more as we carry on. And very crucial, the small intestine is approximately 6 meters long. Maybe you need to measure and see how our small intestines are so long. But because you can see that they are so intertwined together, they are folded here as you look outwardly. They are very long. If we were to stretch these small intestines, they were going to give us plus minus 6 meters long. And the reason for this length is that the length of the small intestine ensures that food remains for a long period, allowing enough time for maximum absorption. So as the food is moving from duodenum to the jejunum to the ileum, because it's quite long, food is taking so much of long time that the process of absorption, the taking in of the food that was broken down by the process of digestion is maximized, because we're talking about maximum absorption. We cannot rob the body. The body must get what is due to it. So we are going to have what we call maximum absorption of the food substance, right? Nutrients. Check this out. This is life sciences is interesting and it's simple. It is so simple. I like it because you learners are very positive. How you like sitting in your life science class, because it relates to your body. And when you talk to your friends or you talk to your families, you are able to relate to what you learn in your class with your daily life. Wow, this is beautiful. We are indeed scientists. Just look at this quickly. So we have got that general structure of the human alimentary system, right? We're not really worried about the presence of the esophagus. We're not looking at the stomach. We are looking at the small intestines. You'll see. So they are so many. Like they are nicely pegged. Nature is amazing. Like somebody pegged them in a specific sequence. And imagine when the food is moving beautifully from the duodenum to the jejunum and moving and moving and moving and moving until we reach the ileum of the small intestines. But look at this now. This is just a portion of the internal part or structure that is showing us the small intestine. We are now moving from the gastrointestinal tract, because that's the entire tract. Then there's the small intestine. There we have the small intestine. And there are different layers that are found in the stomach and also in the small intestine. You can see that we've got muscles that are found in the inner part of the small intestine, right? So as we look at this, the peristalsis is also happening. The peristalsis is also happening, which is going to allow the process of digestion to take place fully so. So what we have here is the small intestine, and we've got different layers. The submucosa is the inner part of the small intestine. We've got mucosa, and inside we also have got the serosa. But we can start relating to this diagram. Don't worry about this diagram. We're going to tap on it so much. But for your information, this is now a fold of the intestinal lining, which is now the villi, or villus if it's one. You will see that there's the different parts that we are going to tap on, but see that, can you see the sequencing from the gastrointestinal tract to the actual small intestine to now a fold of the intestinal lining, which is now having or giving us the villi, as many of villas as one, and we're moving now to tapping into deeper or more detailed parts of the villas or villi, as many. Beautiful. Here we are. So we are looking at the different. You see the folds. It's folded. It's so much. It's folded. You can see it's fully folded. There we are. It's fully folded, and then we have got the presence of the villi, and the villi is made up of a single layer of cells. You see a single layer of cells, which is called epithelial cells. A single layer of epithelial cells. And then now this is also on that thin layer, one layer of columnar epithelial cells. We've got gallbladder cells. We'll talk about the gallbladder cells, but check this as well. We have got blood capillaries, right? So this is basically the structure of the villus. We've got lymphatic vessel that gives rise to the lacteal system. You see the link of the lacteal system with the blood vessels that are found on the villi? This is interesting indeed. Let us talk about the structural adaptation. Epithelium, that single epithelium, you see this is our single epithelium cell. It has got many mitochondria. You know mitochondria, you have learned it earlier on. Mitochondria, it's providing or supplying energy for active absorption of nutrients. So microvilli further increase the surface area. We've spoken about that. So if you look, I promised you that you will see the microvilli. There we are. And we've got blood vessels, rich blood vessel supply. We've got smooth muscles. We've got serosa. Serosa is just part of the serosa is part of the membranes that are found on the small intestines. Checking the epithelial cells, we can safely say that it's one cell thick, so it's really thin, and nutrients can pass in quickly. However, the gallbladder cells, what they do, they secrete mucus, and the mucus, what it does, it keeps the surface moist, right, and allow nutrients to be dissolved and then to be absorbed. You see, the presence of gallbladder cells and the thin lining of the epithelial cells, they play such a role. They help out in the process of absorption. There is a lymph vessel that we spoke about. So we have got a lymph vessel. I've mentioned the lymph vessel here, and which is called the lacteal. You see, yes, the lacteal. This is tiny lymphatic vessels. So they are like capillaries. They are very small, right? And they absorb fatty acids and glycerol. Fatty acids are the product of what? Of a fat molecule after it's completely digested. And now the thin epithelium cell, one cell thick, to increase the diffusion rate, and microvilli increase the surface area for absorption. Food is being digested. And we've got the blood capillaries. We do have our blood capillaries. They absorb glucose and amino acids, right? Glucose, products of carbohydrate digestion, and amino acids, products of protein digestion. Let's talk about the absorption process. Digested food is now soluble form and ready to be absorbed into the bloodstream. And most absorption takes place in small intestine because most digestion has taken place by the time the food reaches the small intestine. Food particles in small intestines are therefore small enough to be absorbed. Hence, we have got the simpler form of the food molecules. I said to you that I'm going to speak on different types of how food is taken in. We have got active absorption. So active absorption, it requires energy for the nutrients to be absorbed against concentration gradient from low to high. So when we've got low concentration and substances must move to high concentration, there's a need for energy. You have learned about this energy, or you learn about it in cellular respiration. But passive absorption is going to take place, does not require energy because it's movement from high to low levels. So there are two different types, active absorption and passive absorption. Just remember that. Glycerol and fatty acids, this is not something new. The end product of lipid digestion, fat digestion, are absorbed by diffusion into the lacteals. You must know where it is going. Aided or helped by the presence of the bile salts, right? This is the lacteal. We have just looked at the structure earlier on. However, we have got other forms of foods. We've got vitamins, mineral salts, which are vitamins are passively absorbed aided by a carrier molecule. Do you know the carrier molecule? It's ATP. Mineral salts are both actively and passively absorbed into the capillaries, blood capillaries. Water is absorbed into the capillaries by osmosis. Just remember, osmosis, diffusion, active absorption, passive absorption. These are the key terms that you need to know when you tap on absorption of food. Then, glucose, end product of carbohydrate digestion, is actively absorbed against the concentration gradient into the blood capillaries. Amino acids, they are the product of protein molecule digestion. So they are actively absorbed against the concentration gradient into the blood capillaries. Can you see? Once food is in the our digestive tract, must be absorbed out of whatever is needed in the blood must go to the blood. You can see that we are mentioning end products of carbohydrates, end products of proteins, end products of fat digestion. Digested, it's now absorbed into our blood system. I want to just make you be familiar with this. Amino acids and glucose are absorbed into the blood capillaries of the villi in the small intestine. Then, these capillaries that we're speaking about, they join together to form large venules, right? These are veins. to form hepatic portal vein, which is now, if you check the diagram, the diagram next year, we've got the hepatic portal vein, right, which is now linked to the liver. Very important for you to take a note of. Transports amino acids and glucose to the liver, right? So the hepatic portal vein is going to transport now what was digested to the liver. And check this out, glucose and amino acids, they flow through the hepatic vein to the heart. So they go through the hepatic portal vein to the liver, and now out through the hepatic vein, not hepatic portal vein, but to the hepatic vein to the heart. And the liver converts excess glucose to glycogen and stores it. This is the lesson that you are going to go through. And excess amino acids are deaminated by the liver to form. The liver plays such a role to form urea. Urea, which is a waste product, can be passed when a excretion process takes place. This table here is showing us the different parts of the body and the types that is going to take place. So we've got glucose digestion, we've got amino acids, we spoke about that, we've got glycerol and fatty acids, vitamins, minerals, and water. So you need to know whether it's active absorption, it's diffusion that is helping, or it is osmosis, or it is passive absorption. You can see that we have got active, passive absorption, and each one there is an indication of what type is taking place. Let us look at assimilation. Now, when food is in the blood, it must go to the tissues. Assimilation is the incorporation of absorbed nutrients into the cells of the body. The body cells absorb required nutrients which are necessary for building and maintenance of compounds. For example, muscle cells. Look at this. Muscle cells will absorb amino acids to be converted to proteins and glucose, which will be absorbed by cells to provide energy. Hence, I spoke about our ability to walk and do different activities. The liver plays such a vital role in the process of assimilation. So the liver is responsible for the metabolism of glucose and the amination of amino acids and the breakdown of alcohol, drugs, and hormones. I even promised you we will speak about hormones. So the liver is having it very difficult. That is why a person that suffers from liver condition, sometimes it's very difficult for the body to be functioning, because the liver will detect and try to render any harmful substance that can be harmful to the body to be harmless, so that we're able to survive. You see, we've got the amination of the amino acids, we've got metabolism of the glucose. If you check this, it's now transport of absorbed nutrients, which you can tap on. The remaining glucose and amino acids, what happens to them? They leave the liver. Okay, so they leave the liver, right? And then they leave the liver via the hepatic vein and transported to the inferior vena cava, to the heart and to the rest of the body. The body needs all these substances. They must be there. And a large portion of glucose is transported to the tissues, especially muscle tissues, where it is broken down by cellular respiration to carbon dioxide and water with the release of energy. This is just something to remember, that cellular respiration, you know the definition? That is when there is the formation of what the body must do. And what is it that the body must do? It must have all the necessary substances that must be carried, and the process has got the products, and the products that we speak about, I just mentioned earlier on now. The products are CO2, and we've got water, and in the process there's ATP that is formed, because ATP is the energy carrier. I hope you are grasping this. And absorbed substances transported, we have dealt with this. You know that we have got fatty acids, glycerol absorbed to the lacteals that we know, and then the lacteals unite, they form lymphatic vessels. I've spoken about that as well. And this, you must just look at the thoracic duct and how they form part of the lymphatic system. Just tap on this as well when you do your revision. Egestion, removal of waste products from the body. Now we've got roughage, which plays a role in digested food substances. What they do now, they are removed from the body, right? And roughage plays a role in promoting peristalsis and prevents constipation and softens the feces, so that defecation, the process of defecation, is safely facilitated. This is just a simple outline of the digestion process. Muscles will contract, and we are going to have muscles relaxing. Circular muscles and longitudinal muscles, they relax and contract, and anus will cause feces to be expelled to the outside. Hence, we're looking at the process, and the process is called defecation. And this, learners, brings us, I want you to go and tap on the different activities, which we are not going to be looking at now, but then we have got activities that you must look at, right? Okay, right, digestion, we're there. These are the activities that I want you to look at. We've got 1, 2, 3 different activities that you must look at, and those activities are going to take you through the process of digestion. And thank you so much. Right? So the activities here, one is indicate whether each of the descriptions in column 1, all right, let's go back here, right? So indicate whether each of the descriptions in column 1 applies to A only, B only, both A and B, or none of the items in column 2. Write only A only, B only, both A and B, or none next to the question number. Can I please advise learners to be very cautious? You need to be extra careful when you answer this, because learners take shortcuts. They will not check on the actual way of answering this. Beyond reasonable doubt, your examination paper will have, this is normally placed at question 1.3. You are going to be expected to answer mix and match questions. 6 marks maximum. It's easy to go questions. So let's have a look at 1. The substance that needs to be digested before the process of absorption. And what is that? A substance that needs to be digested before the process of absorption. Let's see if you have got the answer. Right? The answer is none for 1.3.1. Let us look at 1.3.2. A lymph vessel in the villas of the small intestine. I think you remember that vessel that we spoke about. It is running right at the center of the villas. It looks like this, and it goes down to the lymph vessel. And let's see what's the answer there. By now you know. I think I know everybody knows. Ma'am, I can tell you what it is. Okay, let's see. It is the lacteal. Somebody might be asking, oh, lymphatic nodes. This, they have to do with how the body defends us from infections. That's the lymphatic nodes. They swell up to fight any form of high levels of infections in the body. But the lymph vessel in the villus, it's called the lactea. The next question is the enzyme secreted by the pancreas. The enzyme secreted by the pancreas. Do you remember the tip I gave you? Here now you must tell us which ones are they. Is that proteases or carbohydrates? Which ones? Do you remember what I told you? So let's see if you got the answer right. Think and write the answer down. Please do not cheat. Right? So let's see. Both of them, they are secreted by the pancreas. So the tip I gave you about the intestinal juices and the pancreatic juices, all of them, they are secreted by the pancreas and they are found in pancreatic juices. The three, the lipase one, the protease one, and the carbohydrates one. All of them, they are secreted by the pancreas and found in pancreatic juices. And we also have got the three of them as well. They are secreted by the intestines and they are found in the intestinal juices. Then let's look at the next question. The structure where chemical digestion does not take place. The structure where chemical digestion does not take place. Do you remember when we did chemical digestion? Can you relate to where chemical digestion is taking place and where it does not take place? Look at this. Can you close your eyes and remember where they are found on the alimentary canal or digestive tract? Esophagus, large intestines. Do we have any form of chemical digestion in these two structures? Let's see if you got the answer right. Both of them. Chemical digestion does not take place in the esophagus. In the esophagus, that's where we've got mechanical digestion. That long tube that is running from the pharynx going down to the stomach. Only mechanical digestion. Peristalsis allows the food to move. It's the process of peristalsis only, right? So there's no chemical digestion. There are no enzymes there. Large intestines as well. Large intestines, that's where we've got basically, it's just the absorption of mineral salts and large amounts of water. Absorption of that. But we do not have any enzymes found in large intestines because by the time food reaches the large intestine, food is moving to the descending colon and to the rectum, which is now going to lead to the anus. And the process, egestion, will take place. Egestion that we spoke about. Let us look at this diagram quickly. You are by now familiar with this diagram. We have spoken about it a lot. Remember, when you are in the examination room, you need to do what? You must be able to label your diagrams. You must be able to label your diagrams so you can tell us. I showed you now the lacteal system. Yes, the lacteal system for us. Oh, let me use the red color. Okay, here's the lacteal system for us. And here we've got the thin lining. You can remember that. I'm also checking this out. Do you remember what this is? See? Do you remember? Definitely you should remember. Okay, that's fine. However, let's answer the question. The first question says, label part A and E. So we are going to label part A and we are going to also label part E. But whenever in the examination, you need to quickly label your diagrams so that you know what you are supposed to label, right? And you can answer the questions that are asked later. So can you tell me the labels? Okay, fine. Let's see if you got this right. Oh, brilliant. A, it's our columnar epithelium. Can you tell us something about the suitability of these columnar epithelium cells? Can you tell us? It's that single film. It's a single, it makes it not thick but thin for easy absorption. So it's columnar epithelium, that's A. And let's look at E. E, these are the goblet cells. By the way, goblet cells, what do they do? Yes, we are not asking this question, but you can tell us what they do. Goblet cells, what do? They secrete what? Mucus. And mucus is going to do what? It's going to keep the area in the small intestines moist, right? And nutrients, they dissolve in it for more maximum facilitation of the process of absorption. So just remember, and when you look at this diagram, can I give you a tip? Something that you can do when you answer your, not even answering, when you're preparing for your exams, or just, this is just a tip of keeping your notes. You can have what we call annotated diagrams. So, okay, fine. You know about annotated diagrams, right? Annotated diagrams, right? Diagram or diagrams, depending. So what you do on this diagram, for example, you are going to say, this is the B, maybe you'd say that is a lacteal, or you are going to tell us about goblet cells, and you indicate that they secrete mucus, and what is the function of the mucus. You can tell us about columnar epithelial cells. You can tell us about the blood capillaries that are found around the villi or villas, and you can talk about the lacteal system. Easy. This is easy. There's no way you can get confused, right? So, let us see. The next question says, briefly explain the function of structure B. Here we've got structure B. We want to see the function. You briefly explain. Can I give you a tip? Just for your information, this has been said so many times, but learners still lose marks because they miss out on the doing word, which is a verb. Here the question says explain. Sometimes the question will say state or name or describe or define. You need to underline it so that you know what the examiner is expecting you to do. Yes, you might have studied the entire textbook, but in order for you to tap on what the examiner is asking, you need to check the verb that is used. Key for you. Know if the question says explain, you are able to explain. Know if the question says state, you just state. And also check mark allocation. Your examination, as you practice for writing your exams, you will see that the examination papers, they've got mark allocation for each question. That gives you a clear indication of how far you can go with answering your question, or how less you must answer your question. Some learners, they will write the whole paragraph for 1 mark. And unfortunately, if the principle of that examiner says mark the first one only, you even wrote in bullet form, and your correct answer is the 4th bullet. So mark the first one only, the first one is incorrect, and you lose marks. So be very assertive when you sit for your examination. Even when you practice, every time you practice, you must check your mark allocation. So we are going to look at the explanation of the function of structure B, right? Okay. So structure B, absorbs glycerol and fatty acids, which is by diffusion. You see now, even the process itself, diffusion. We spoke about active absorption. Somebody is shouting out the other one. Once I mentioned active absorption, somebody said, I know, ma'am, we've got passive absorption. All right, yes. And what else do we have? Osmosis. But now, part B is responsible for the absorption of glycerol and fatty acids. By the way, what is glycerol and fatty acids? These are the simpler form of the digested fat molecule. And what enzyme is responsible for this? Is the enzyme lipase. And where do we find it? We find it in pancreatic juices, which is secreted by the pancreas, carried by the pancreatic duct to the targeted area, and also intestinal juices secreted by the intestine. But check this out. This is aided. The process of diffusion is helped by bile salts. If you check one function of the liver, it is to secrete bile. Oh, I did not mention this. The relationship that we have, we've got the gallbladder and we have the liver. Do you know that learners have got a misconception? They think that the gallbladder secretes bile. No. The liver secretes bile. And the gallbladder is the storage space for what? It is the storage space for bile that was secreted by the liver until there's a need. In fact, I should have mentioned to you that when we were talking about the process of absorption, when we've got excess of glucose, this is going to be converted. Excess, anything that is in excess, that's something that the body does not need. And once it is in excess, it means that it must be removed. But we cannot remove, the body cannot say, some of the things, yes, they can be ingested or removed out of the body. But there are those that will be needed later when the need arises, like glucose. There's no way that glucose can just be discarded out of the body. So excess of it, we need, hence, we have spoken about, or you have learned about homeostasis. Homeostasis. This you must remember because this apply homeostasis. Just remember this because this applies to so many things. Our normal functioning or the normal functioning of our bodies is controlled by homeostasis. The body has a way of detecting if something is more or something is less. So if we've got more, it means that the body must tone down to become normal. And if it's less, the body must find ways of taking it up. I don't want to tap more into this because you will learn about homeostatic balances in our bodies. But keep this in your mind that if we've got excess amino acids, they cannot just be excess. And that's fine because then that can become uncomfortable and harmful for the body. And if glucose is more, excess can at least have, the body has a way of saying, I can store this until a need arises. And then that excess glucose will be converted to be in a user-friendly way. And then it can now be used by the body. Right? This other question is saying, name 2 substances, right? Here you are just naming. So you are naming 2 substances which are found in higher concentration in C than D. Higher concentration in C than D. Can you check something here? So movement of nutrients is taking place. You see this arrow here is leading us into the villas because this is one. And in the process, there's absorption taking place, right? But now, as we carry on, we move, and here we find C, which is leading us to the exit of the villas. So you name 2 substances which are found in higher concentration in C, right, than D. Okay. Right, let's see if you have the correct answer. And let's check this out. So in C, we've got large amounts of glucose and amino acids. Large amounts of glucose and amino acids. They are found as they are getting absorbed. We've got large amounts of glucose and amino acids. And you have learned about how the body later is now speaking to the liver system, excess of glucose, which will be converted for storage, right? And that has to do with the liver and the muscle cells, right? And the amino acids, if they are found in higher concentration, we are going to have that process which we call deamination. We've got the process called deamination. And now, let us look at another question. The next question here is, explain 2 reasons why this structure is so efficient in its function. So we are looking at, firstly check the doing where is the explain 2 reasons why this structure. What is this structure? This, who can give the caption of this structure? By the way, when you are expected to draw, you will be drawing. I'm not saying you will be expected to draw the structure of the villas. I am saying that in life sciences, whenever you are expected to draw something, you first start with the caption. And what is this structure here? Who can say or who can tell? Okay, yes, this is the villas. So you must write it as a structure of the villas, right? It's a singular form. You can even say villi, right? So we are going to explain now. Explain 2 reasons why this structure is so efficient in its function. If we say that, oh, that learner is efficient, if you give a task to this learner, to Tabang is very efficient. It means that Tabang will carry the task that he is given to its completion so well that there will not be gaps. But now we are looking at our bodies. Why are we saying the villi are efficient in their function? And you know the function, right? So let's look at the possible answers that you can give. Right. Okay. So it is well supplied with blood vessels and lacteals. The presence of lacteals and the blood vessels, you can see, oh, I did not mention this. Let me use my red pen here for you to see. Can you see that we've got blood vessels that are richly supplying the structure of the villas, but you see the capillaries, they are intertwined here, the capillaries. See the close relationship that we have here, right? And I'm going to show you in this color here. This is the lacteal. You can see the lacteal system is here inside. Right, there's the lacteal system. But can you see that as I start to show you how closely linked the structure is, the blood vessels. So we are saying that it is fully supplied with the blood vessels, right? So it's fully supplied with the blood vessels and the lacteal, which is going to assist to transport absorbed food nutrients. Because anyway, there must be transport system. Food nutrients must be transported. And it is moist and has mucus. We just mentioned earlier on the presence of mucus. Which structure secretes mucus, by the way? Who can tell? It's gallbladder cells, all right? So we remember, yes, we remember about gallbladder cells. Gallbladder cells, right? And now they produce the mucus, which keeps the area moist, right? Look at the link. To facilitate or to speed up absorption for better diffusion. Because in a moist environment, the movement of substances from higher concentration to a lower concentration, it's even better. It's faster. It's efficient, right? Somebody is asking me, Madam, is it necessary for us to mention that it has got gallbladder cells? Yes, you can mention that. It is fine. That shows us that you know your work well. And then let's just quickly clean up here so that you don't struggle with your work here, right? Okay. Okay. So we're now going to speak about the presence of the folds that further increase the surface area for food absorption. Okay, so let's have a look. I want us to look at that now. Right, okay, right, doesn't matter. So we have got the presence of many microvilli to increase the surface area for food absorption. We said that the villi has got microvilli and the surface area is increased. And once the surface area is increased, we are going to have better food absorption. And check this as well. It is thin. That single film of columnar epithelial cells, right? They allow the digested food to be in close contact with the blood. Thin, so the digested food is very close to the blood. For what? For allowing the process of absorption to take place, and contains many mitochondria. You have learned about mitochondria. This is the powerhouse, is the center for energy, for active absorption of nutrients. Hence, when we speak about active absorption, you know that active absorption energy is needed because this is against the concentration gradient. So absorption is taking place from a place of higher concentration, from a place of lower concentration to a higher concentration. Therefore, there's a need for energy to allow the nutrients to be absorbed, right? And so you're fine. Imagine if the examiner can give you a question with 8 marks. You score them easy. Okay, you just mention the presence of blood vessels and the lacteals, and you tell us to transport the absorbed food nutrients, and then you say, oh, by the way, there's something called moist. Okay, yes, I got it. Gallbladder cells, they secrete mucus, which keeps the area moist and then allows the nutrients to dissolve for better diffusion. You can see the mark allocation as well. So you tell us the structure and you tell us its function. Like you speak about blood vessels and lactea, then tell us what they do. They transport the absorbed food nutrients. You speak about the presence of mucus, which is secreted by the gallbladder cells, and what do? They facilitate or speed up the process of diffusion. And the presence of many microvilli, and what does it do? To increase the surface area for food absorption. And thin, and which is going to be now the presence of the one film layer of columnar epithelium to allow digested food to be in close contact with the blood, and contains mitochondria, which is for active absorption. Quickly, just remember that we spoke about osmosis, movement of water molecules from a higher concentration to a lower concentration. And we spoke about diffusion, which is the movement of substances from a place of higher concentration to a lower concentration. That's diffusion. And active absorption is the movement of substances which are now moving from the active absorption from the place of high concentration, from a low concentration to a high concentration, against the concentration gradient. And now passive absorption is the movement of substances from this one is easy. It's osmosis basically, from a lower concentration to a higher concentration. Where we need more energy, it must be the presence of the mitochondria to provide energy. Right. Are we happy? We find our footing, we are fine, we are good, and everything is fine. So I want us to quickly go back and to recap things that we are supposed to recap. Right? Maybe on this note, let's look at the completing the blanks, which are found on the table. We have looked at this table, and let's see that we have got a process. So absorption is taking place, active or passive absorption is taking place, and we have got the structure. So it's active or passive absorption, right? Let's go, we're going back there. Okay. Right, must not cheat. So active and passive absorption, we've got structure where absorption is taking place. So we've got glucose, digestion, we've got amino acids, glycerol and fatty acids, vitamins, minerals, and water. Right. I wanted us to just go back and tap on what is producing the end product of glucose. Who can remember? That is produced by glucose. Comes from what? Glucose comes from what? Is it carbohydrates or is it protein? Somebody knows about it? Glucose is the end product of carbohydrate digestion, right? And amino acids, they are the end product of what? Of protein digestion. And glycerol and fatty acids, these are the end products of the lipids or the fats molecules, right? So vitamins, minerals, and water. And this table here, once you know it, you are able to tell what is happening where. So let's have a look at our possible answers. Right. So absorption, active absorption or passive absorption. Let's check. Glucose. What kind of absorption is taking place when glucose must be absorbed in the body? It's active. So it means that it is moving against the concentration gradient. And the structure where absorption is taking place, this is taking place in the blood capillaries. Do you remember the blood capillaries that we saw on the structure of the villus? That is exactly where absorption is taking place, right? And we have got amino acids. Amino acids, just to refresh your mind, amino acids. Yes, it was that whole band of amino acids broken down into amino acids from the whole full protein molecule, right? So amino acids, what kind of absorption is taking place? Absorption that takes place for amino acids is active as well against the concentration gradient, right? This is taking place against the concentration gradient, and structure where absorption is taking place, it's also in the blood capillaries. Blood capillaries. We've got lacteal system, which comes from the lymphatic vessels, but we've got blood capillaries. So just remember that glucose, amino acids, both of them is active absorption, and then absorption is taking place in the blood capillaries. But if we check, or if we look at the glycerol and fatty acids, right? The glycerol and fatty acids, right? Oh, at least my pen is not striking anymore. Right, so we said that active absorption here, it's for the glucose, and it is taking place by the blood capillaries. Amino acids, active absorption, and it's also happening at the blood capillaries, right? I'm going to use red, which is going to show better. Okay, let's see. Just quickly clean here. Right. Good. Okay. I was still saying that we have spoken about this fully, but for your information, know that absorption is taking place by the blood capillaries, and this is for glucose absorption. I am going to show you again, I'm going to show you the actual diagrams, because now when you meet the actual questions, some learners are saying, oh, this is quite confusing. I don't understand a little bit. Come again. Maybe you were too quick. No, that is fine. Never mind. Amino acids. Now, amino acids is active absorption. You have to choose active or passive absorption. So amino acids is actively absorbed. This is against the concentration gradient. Right. And it is taking place by the blood capillaries. Happy as. Glycerol and fatty acids. Right. This is the simpler form of the fat molecules that were broken down. Fat molecules. Right. Glycerol and fatty acids. And this is now, the process is passive diffusion. Passive. It's not active, it's passive. Remember, diffusion, movement of substances from a higher concentration to a lower concentration. There's no need for energy. Let me just give you a simple illustration here. So if we have, I'm just going to give you a simple illustration. So if this is high concentration, right, diffusion will mean that substances are going to move from their place of a higher concentration to a lower concentration. So movement will take place from this higher concentration. Let's say we've got this. So the movement is easier from higher concentration to a lower concentration. However, when we speak about active absorption, for example, I'm going to give you another illustration. Let's quickly clean this. Right. Okay. So if we have this, right, so this is our low concentration, right, and then we've got our high concentration here. Can you see that the movement is not easy? For substances to move from here to here, it's not easy because it's from the low concentration to a high concentration. So as the substances move, now we need energy, right? We need energy. And this is now against the concentration gradient. But let's check vitamins. Vitamins is both. Vitamins can be actively absorbed or passively absorbed, right? And where is the absorption? It's taking place by the blood capillaries. Minerals, right? Minerals also, it's active or passive absorption. And where is this taking place? It is taking place by the blood capillaries. Water is passive a process, which is called osmosis. Hence, the main thing of all the four, it's here on this table. Osmosis, diffusion, here is diffusion, I spoke about it. And then we've got active, and the passive ones, it's the two examples, osmosis or diffusion, right? So water moves through the process of osmosis to the blood capillaries. There's something that I want you to check up. Let's just fix what has been bugging you, right? Let's check this out quickly. The diagrams are there. I hope as you answer the questions, you are able to see where you are found wanting. But I want us to quickly look at the structures again. Digestion is fine, but what might be giving you issues is right. We are checking again the same structure of the villas. We are recapping now, right? So you see, we spoke about, now you are going to relate even better, because you see what I was telling you. You see, when we speak about the efficiency, it's the thin epithelium, right? Thin epithelium, right? Which is one cell thick to increase the diffusion rate, structural suitability. And the presence of microvilli, right? There's your other mark, to increase the surface area for the process of absorption. But now, lacteal, right? Lacteal, here's the lacteal system, which comes from the lymphatic system, right? Lymphatic vessel, right? So the lacteal, there are tiny lymphatic vessels, and what they do, we just spoke about it now, to checking the table here, which is relating to what we answered now, right? So it's for the absorption of fatty acids and glycerol. But if you check, we've got blood capillaries here. They specialize in the absorption of glucose and amino acid. I hope that confusion of not relating to the structure, you can see the thin film of the epithelium cell that is going to give us the nice, easy area for the diffusion of nutrients. I also want us to check quickly the following. Right, so let's check this out as well, because I saw that you were right. This is okay. Let's have a look quickly on this. Right, just check this out. Glucose, as I have asked you many times, it's the end product of what? Of carbohydrate digestion. Do you remember the name of the enzyme that is responsible for this process? We'll tap on it again. It's actively absorbed against the concentration gradient. Learners get confused. That's why I kept talking about active absorption and passive absorption. Right. So carbohydrates into the blood capillaries. Here are the blood capillaries. Let's see. So we are talking about glucose. Yes, we got it right. And then amino acids, the end products of protein digestion, are also actively absorbed against the concentration gradient into the blood vessel. So glucose and amino acids. But as per indication, we speak about lacteal, but lacteal, the main focus, is fatty acids and glycerol. I want us to also look at the different, I want us to look at the table that we had. right? Let's look at the table that we had earlier on. We're getting there. Right, this one. Okay, if you get confused with this, you have missed your digestion story, right? And just want to, okay, let's quickly get in the duster and we erase what is here, what we don't need. Right? Let's see. Let's see. Right? Okay. All right. We're checking that out. Okay. I don't want us to look at the diagram of the human digestive system. I just want us to refresh on this. Right. So it's fine. So we said that proteins. So once you have looked at mechanical digestion and chemical digestion, now you concentrate on what is broken down to become what at the end of the day. So we've got proteins, we said, and the name of the hormone is protease, okay? And protease breaks down protein molecule to become what? Amino acids. You see, amino acids are broken down. We are just recapping what we have learned because I can see the gaps of learners getting a little bit confused and edgy about how must I answer this. Now, Mami spoke about intestines. I'm confused. There's assimilation, there's absorption. I don't know what you're talking about, right? So here we've got proteins which are broken down by the protease. Check the spelling. It is key. Learners have a tendency of asking, ma'am, does spelling count? Yes, spelling counts a long way, right? So amino acids are formed, and this is the end product. Hence, later, when we look at the diagram of the villi, we're talking about where the amino acids are being absorbed in the small intestines, right? They are absorbed in there to the blood capillaries. And starch molecule, one small starch molecule, it's the carbohydrates. Carbohydrates, what is carbohydrates? Carbohydrates is the name of the enzyme. So if you're asked about enzymes and you say carbohydrates, you are incorrect. Carbohydrates are food molecules, but not an enzyme. And we are looking at what is the end product. When starch molecules or carbohydrates broken down by the enzyme carbohydrates, we are going to have glucose molecules. Just check this. Can you see the amino acids? They are in a simpler form. Look at glucose. This is now in a simpler form. And our fat molecule, here is our fat molecule, right, which is made up of fatty acids and glycerol. But look, check this out. This is like one complex structure. And lipase as an example breaks down the fat molecule, and when it is broken down, it is, can you see now the end product? Just check this. This is such a simplified diagram, right? You can see that glycerol, fatty acids, they are now no longer one complex unit because lipase had worked or broken them down. But everything here, it is chemical digestion. Enzymes involved, chemical digestion. And as per indication, now you just tell yourself, I know by now the three different enzymes, protease, carbohydrates, lipase, common areas where they are produced. Pancreatic juice, you see it's pancreatic juice here, and then pancreatic juice here, pancreatic juice here. And also intestinal juices, intestinal juices, intestinal juices. Just remember what I said earlier on. Only carbohydrates is also found in the saliva, right? But we will not find protease and we will not find lipase. And in the stomach, we only find this protease as the enzyme that helps to break down food in the stomach. Let's just check something else as well. Things that you were worried about. Okay. There was a question that we have answered which said which two parts we did not have chemical digestion. So, and I said to you, but I was just explaining to you, hoping that you still can recap and you can remember how the structure looks. But let us check this quickly as well, right? So we have got our alimentary canal, basically that is the overall. Okay, that question was asking about the esophagus and the large intestine. So here's the esophagus. As I showed you that this place here, it's only muscles. It's only circular and longitudinal. Circular and longitudinal muscles, which do what? They do contraction and relaxation. They contract and relax. And as they contract and relax, they allow the food bolus to move. So the food bolus is going to be basically inside the esophagus, will be moving, going down. It will be going, the food bolus is going to be going down, right? So there is no chemical digestion. And I said to you, when we look at the colon, this is our colon. This is the colon. Ascending, transverse, descending. There is no chemical digestion taking place here as well. However, the stomach, where we've got enzymes that are secreted, they help in breaking down of food in the stomach. We also have got the presence of hydrochloric acid, which is going to help to digest food or to break down food. We also looking at the small intestines, we are going to have our chemical digestion by the presence of enzymes. But in the mouth here, by the salivary glands, these are the salivary glands. I mentioned to you how they are stimulated to secrete saliva because they are stimulants, they are triggers that can trigger the salivary glands to secrete saliva, right? So here we've got, somebody must know the name of the enzyme that is found here, and then you must know the enzymes that are found in the stomach and the ones that are found secreted by the pancreas, right? Right? So in the pancreatic juice, by the pancreas, we will have enzymes, the three enzymes, and the intestines, intestinal juices, right? So we're speaking about intestinal juices, right? And all these juices, they contain enzymes, and the enzymes, what they do, their responsibility is to aid or to help with the process of digestion, right? So basically, even from what you learned last week, you know there's ingestion, taking in of food, and after food is ingested, taking in. After food is ingested, then we have got the process of digestion. That chart diagram that I showed you, we've got digestion of food, mechanically and chemically. Mechanically, mastication in the mouth, food is chewed again and again and again, food bolus is formed, the tongue helps the food bolus to be swallowed. Peristalsis movement, hence you must know the meaning of peristalsis, pushes the food bolus and the food bolus starts to move along the alimentary canal, right? Mechanical digestion into the stomach, and then the continuous churning in the stomach will help food to be mechanically digested. And when digestion mechanically is completed, we have got now. And remember, it's not like one form of digestion must take place and the other one must wait. They happen simultaneously, like in the stomach. As the stomach churning is happening, that is mechanical. We will also have got chemical digestion, where food is going to be broken down into simpler substances. So just take a note of that as well. And chemically, I've really drilled you through chemical digestion. There's no way that you will forget proteins, carbohydrates, fats. You will not forget that they are broken down by this enzymes, and then you'll also remember their end products. Good enough. Once we have that completed, you know that there must be absorption. That is when now the structure of the villas comes into the picture. And you will tell us, by now you know the structure of the villas. We have done it so much. You look at how it is efficient. Now you look at the presence of the single columnar epithelial cells, which allow for easy and fast diffusion of substances. You'll speak about the presence of the goblet cells. And goblet cells, what they do, they secrete mucus. And what is the purpose of mucus? It makes the area to be moist. Remember, diffusion happens easier and faster in the moist environment. It is moist, and also it is going to allow now the dissolving of substances. All right? And we will also speak about the presence of the lacteal and the presence of the blood vessels, right, which are now going to allow the absorption. And don't forget that amino acids and glucose, these are absorbed in the blood capillaries, and the lacteal will absorb the glycerol and fatty acids. Once that is finalized, we're looking at egestion. Egestion, we spoke about roughage. So what do we eat helps us in the process of relieving ourselves when a defecation is taking place. Removal of undigested and indigested food material from the body to the outside, right? So that is egestion. In fact, before egestion, we've got assimilation. When substances are in the blood system, some nutrients, they need to now get into our body tissues so that we're able to perform our everyday processes that I have mentioned to you. Processes like running, like talking, like you are listening and participating in this class because you are energetic. So we need the presence of nutrients in the cells beyond the blood system or beyond the blood capillaries. And then now the closing process is egestion, where food is being removed out of the body. I also mentioned homeostasis. Do you know that if you don't eat enough roughage, some people's digestive tract are quite stubborn. So you experience constipation. Constipation means that there is no harmony in your body. There's no homeostasis. Whatever is indigested must come out. And that is why I'm not going to speak about the nervous element that is found on the rectum to indicate to you, to your brain, because this is now grade 12 work, to indicate to the brain that you are pressed, you need to relieve yourself. But remember, when you are pressed and you want to relieve yourself, this is something that you don't decide. I want to be pressed now. It comes as an involuntary action that is taking place in your body. So if somebody is constipated, it means that you struggle, hence there's no homeostasis. As I mentioned to you, excess of amino acids, we don't need excess of amino acids, we do not need excess of glucose. Whatever is in excess either must be converted into a different form that must be used in the body. The same thing applies to egestion. Whatever is not supposed to be in the body must go out. And if you suffer from constipation, then you will not be able to enjoy your next meal. Some people do suffer from chronic constipation, and some people can even experience what we call the entangled small intestines. Imagine if, because it's long, I said it's at least how long? Plus minus 6 meters long. So if it's entangled, and you've got an obstruction. It means that there is no movement of food through your digestive tract and you experience that blockage. It might lead that you have a surgery. So they can remove that entangled part of the intestine and then fix it up so that movement can take place. So the body is well structured to work together from the mouth until the anus, where the process is concluded. I hope that you had a good session, and I hope that you are ready and looking forward to the homeostasis, the balancing of what must not be in the body and where it's supposed to be stored in the body and in which form this is going to be stored in the body. It was a beautiful session. A lot was said today. More details were given. Nobody has a reason to say, I do not understand human nutrition, because whatever you went through made life easy for you today. Taken you through all the little things that confuse learners when they come into the examination room. But remember, when you sit for your examination, you need to be calm, you need to find yourself, and you need to be at peace. Right. And learners, this you have done, this you know, you have done this, you all know this, you have done all this. It's not Greek to you, it's easy, right? We have looked at the absorption, cellular respiration story. We looked at nutrients which are absorbed and where they are absorbed. We looked at digestion, and we looked at the activities that we have answered together, which made it easy for you. I've went back to highlight to you what needs to be done. And we are, on that note, going to say it was a beautiful session. Thank you.
Sorry.
How I survived.
How you survived?
Why, this is torture.
What? This thing?
But it's fine. I'm not upset. I improvised.
You improvised, yes. I saw that. I saw that. Unfortunately, yeah, but
but it's
you
fine.
tried,
Yeah, no, I
Next
really,
time.
yeah, it's you know what? It does not. I don't know. This pen sometimes does not pick up the pen.
Oh,
So you come, you tap. You know, you tap. Sometimes it picks it up when you tap
Yeah.
on the actual. You see.
yeah,
And sometimes when it does not come up. I need to come to the gray area.
yeah.
And you tap, it doesn't come.
It does not come.
You tap, it does not come, and you must. No, it's fine. In the meantime, you must keep moving.
Yes.
You understand what I'm saying? So
Yes,
I don't know what is it, because I have seen struggling presenters.
Yes, yes.
What is it? Is it us, or is it the Apple? Or what is it?
Yeah, I think.
What, maybe it's us.
Yeah, it could be because
Mm.
the problem with these things, like touching the edge, I'll say, okay, you touch in. Oh.
Mm-hmm. You can keep. Mm-hmm.
I wanted.
A slideshow.
So it went out.
Mm.
Yes.
Okay, so we can come to. All right.
I was trying to move it to to get it to somewhere where I can write. Okay, you're writing.
Wayibona?
Yeah.
Then it does not come out. You keep tapping on the area, the grey
Yeah,
area,
the
because you
grey
want the
area,
pen to
yes,
come.
yes,
Because
yes,
here you
yes.
must go to the pen, you must also go to the eraser in order to move. Otherwise, it gives us a problem.
Okay. Let's look at it. And
Okay.
see. Because then some, yeah. Let's look at it
Okay.
and see
What it is.
does, yeah, what it is.
But I get you also saw when you are tapping and you want to go.
No, I know. It actually gives me hassles, but see these guys, then it comes. There's a pen, and you select the eraser.
Can you see how it runs as well?
Yeah.
When you go to the eraser, sometimes you want to erase, ne?
Mm.
Then it moves to the. If you don't know your story, you get so confused. But lucky enough, I knew what comes first, and then I go back to say, never mind, you know. Let's check the pens, the eraser. Try to erase. There it is.
There's the eraser.
Mhm.
Oh, okay, no, I've saved.
Oh, so it will
the
not
rest.
erase.
Yes.
You save the
So,
annotations.
Yes,
Mm.
I have to first write.
Mhm.
Let me just follow how you.
Mm.
Then when I go to
The
the
pens.
eraser.
Mhm. What?
Mm.
It matter to the next one. And o tshwanetse o kgutlela ko morago.
Mm. I don't know if it's dirty or what.
I don't know. But it iyaphazamisa. But it's not a train smash. It's only that for the learners also to see this and then you go back. Shame. But they flow with us.
It's dirty.
Oh, it is, ne?
Yeah. I just want to check if you can feel it with your finger.
Ah, it's dirty.
It's dirty.
Oh, very. Mara wayibona leona
Yeah.
hore. This
No,
taka.
I think
Is that
our guys, they using the wrong thing to clean here.
Oh,
Yes.
can you see it's
If
dirty?
If you look at it,
Mm.
you can touch it. You can feel. So this is the spray.
Mm. Oh.
This is the spray. Okay.
So the spray iphazamisa movement.
No, because it's a layer.
Mm.
So
Adibuyisana.
the pen
And
does not touch the screen because if it lands on top of
a particle.
a particle, it's
Mm.
not.
And then if it
Okay.
touches, sometimes it touches where there's,
yeah.
then they
Okay,
run.
No, because I felt it. It's there.
Mm.
This thing is supposed to be smooth.
Now I was thinking when I started, ne?
Oh.
sorry.
Actually, I've
I was-
not been checking too much because
No, I understand. And now I was thinking maybe ke color ya,
it's
ke re ke
dirty.
color ya yona, ke re hai, mara a ke
It's
tsebe.
dirty. But we'll check.
Okay, that's fine.
It could be that. It could be
Other
something
factors.
else,
Okay.
Yeah, so we just need to check
Okay.
because this should move. I mean, it's even an apple pen. It's not
Mm.
like.
In ordinary, there's those ones that you use to jam.
Yeah.
This one must just move
you
smoothly,
should
nicely.
Just move. When this thing is meant for that.
Mm.
So I will check and I'll try and first is to clean up, but I must show them
Okay.
because I did say we must be careful. with what we are
These
cleaning.
gadgets are very sensitive as well.
Yes.
Wa bona
yes.
gore e
I
latlha
mean,
memory ya
this one-
yona.
This one, it should be as smooth as this.
Mm.
Just touch it with your fingers.
Then
like
it
that.
it moves.
Just move your fingers on top of it.
Aye, they rough.
Yeah. No,
It rough.
just here, even everywhere.
It rough.
It's rough.
It is rough, shame.
Now, this sharp point, when it lands on the
Particle. They are fufa. They are fufa, and then how do fufa?
I hope it's not permanent
Damage. Oh, please. Because why wanna carry iko kamukar? Some places I think it's inside.
Yeah, it looks.
Hmm.
No, it looks like I can take it off. Yeah.
Okay.
Yeah, you
Go
see,
on.
it's
Go
gone
on.
here. It's gone here.
Go on.
But as soon as I move my finger there, but it's gone. So it's a lot of work.
But careful handling.
Yes. Well, we'll have to switch it off and then
Clean it up.
Thank you for that. But
No, it's fine.
We apologize.
No, don't worry. Because lucky enough, because we are, it's the system that if you are a starter, you will die of panicking.
Yeah, you'll die.
Literally dying.
But it affects the whole
Session. The movement of the session becomes. It's all over.
It's all over, yes. I saw that.
Hey, all right. And your mind now, you're missing your touch with the kids because you are worried about the gadget.
And then as soon as I stand up, then you are fine. Then I'm thinking, let me not disturb everything. Yes. And also, I would not have done much because this thing is what it is.
Yes. So we managed.
Yeah, no.
We managed. Thank you so much.
Thank you.
It's not a straight. Don't worry.